Hello students, welcome again for the biology lecture and let us start with the new chapter, that is the molecular basis of inheritance. Molecular basis of inheritance. We know that this is related with genetics. In the genetics, we know the characters are transferred from first generation to the next generation. That is from parents to their offsprings. And these characters are exactly similar to that of the parents. For example, baby elephant having the same characters with its parents, as well as human beings. Children are produced which are also having the same type of the characters which are seen in the parents, that is father and mother. So these things, that is the characters are transformed. Some think our factor is there which is responsible for the transformation of characters from one generation to the next. And we have studied in the third chapter, I think, that the genetics, the pioneer work and the foundation of the genetics laid by the scientist who is also known as the father of genetics, that is the Gregor John Mendel. So Mendel, he performed the experiments on the basis of some contrasting characters, and he selected for his work, that is the plant Pisum satire. In a common word, it is called as sweet pea. This sweet pea or garden pea, where there are so many characters which are having the opposite type of the characters. For example, the height. If you study about the height or observe the height, it is of two types. It is tall and dwarf. So this type of the character which is related with the same type of the character which is called as the contrasting characters. These contrasting characters, that means the characters which denotes the same type of the character which is having in two different plants. That means some plants may be dwarf, some may be tall. Like this. So many characters, about 7 characters are there in the Pisum satire. And Mendel, he studied all these characters, those are about noted. He noted about the 7 characters, 7 contrasting characters. So this pioneer work, he performed experiment and he put forth some laws for us. Those are known as the Mendel's laws. And these laws, along with some crosses, that is a monohybrid cross, dihybrid cross, and so on. So all these things he studied in his experiment, and he concluded that the characters are transformed, and it is responsible because of some factors. He gave the name factor because up to that time nothing was investigated, but only some responsible factor must be there inside the body, inside the cell, and he concluded and gave the name, that is a factor which is responsible. So the continuation of the link, that the factor, so many scientists, they try to discover, investigate the factor which is responsible for the transformation. And this chapter is related with the same one, that is the inheritance, that the character transformed from generation to generation. And many scientists, they tried to go in a deep, that is in the molecular level. You know, molecular level, it is the level where we can see different chemical composition. So this is the chapter related with the molecular basis of inheritance. So let us see the next type of the investigation of the discoveries made by different scientists one by one. So the first experiment performed by Griffith, and Griffith has given us a very pioneer type of the work. Before that, his work, the Frederick Meister in 1869, he worked and he tried to sort out some factor which are responsible for the inheritance. So he worked with the white blood cells from the infected pus, and that is from nuclei. So the extracted material, he gave the name nuclein, and this nuclein is having acidic properties. So it was named as a nucleic acid. Nucleic acids, which are in a high amount of chemical factor, that is a phosphorus. So in this nuclein, phosphorus contents are much more, and because of the properties with respect to this content, he named as acids, and thus it is obtained from the nucleus. It is called as a nucleic acids. So two kinds of the nucleic acids, which are known as the DNA. You must know popular about this DNA as well as RNA. DNA stands for the deoxyribonucleic acid, where one oxygen is less, so it is called as a deoxyribonucleic acid, and RNA, that is the ribonucleic acid. Both are having the same type of the composition or the component, only one oxygen is less in the DNA, and of course it is acidic in nature. In 1900, so many scientists, those worked on the genetic, so they are called as a geneticist, and the factor, that is the DNA, which is called as a gene that controls the inheritance, and it is located on the chromosome, which consists of much amount of protein and DNA. So that is the histone protein, and these chromosomes, they are more conspicuous, or they are more distinct. We can see under the microscope, a compound microscope, of course, and these chromosomes can be seen during the cell division stage, that is from the prophase, metaphase, anaphase, and telophase. Like that, when that is in the condition of the cell division, at that time these chromosomes, they can be seen under the microscope. Even we can count the number of the chromosomes. So the number of the chromosomes, they are certain or specific in each and every plant, animals, and each organism. So the number is fixed, and in this chromosome, these chromosomes are present inside the nucleus. They are membrane bounded. When we are talking about the eukaryotic cell, they are membrane bounded. Histone protein is there. So these genes or the DNA, that is mostly composed two things, that is the protein, and second one is the DNA. So to find out this content, whether the protein or the DNA is responsible for the genetic information, that is the transformation from first to next generation. So what I told before, the Griffith experiment, he performed this experiment and tried to find out the factor or the principle which is responsible for the genetic information. So let us see that Griffith experiment, which is the pioneer type of the experiment that come to the conclude after the experiment. Let us see, or let us discuss about this Griffith experiment. So Griffith, he was the medical officer from the Britain. So it is a British medical officer, and this Griffith, that is a Frederick Griffith, in 1929, he performed experiment, especially on the mice. There is a group of mouse. So he performed the experiment on the mice and he injected some injections like structures that is of the different bacteria. So he chose for that experiment, that is the Streptococcus pneumoniae. In the last book or the previous book it is Diplococcus, now it is a Streptococcus pneumoniae. Streptococcus is the generic name and this is the bacteria. So singular is the bacterium and species. The species name is given because of the disease which it produces. That bacteria produces a disease known as the pneumonia and hence the species name pneumonia. And Streptococcus pneumoniae, this is the bacteria. This bacteria is responsible for the pneumonia in mammals. You must know mammals, mammalian characters. So human beings are also included in the mammals. And pneumonia, you must know this type of the disease nowadays in the COVID-19. Such type of the disease, which is very serious type of disease, where the respiratory tract is totally infected with this bacteria and that person cannot breathe properly, and hence because of the large amount of mucus formation, suffocation is there and it is rather difficult to breathe, and hence that patient that goes in a serious condition and may cause further death. So this Streptococcus pneumoniae, this type of bacteria is selected, Griffith selected, and he observed that there are two strains of Streptococcus pneumoniae. As usual, these bacteria, they are of having different strains. Commonly they are positive, negative like of the strains, but here it is the Streptococcus pneumoniae having two strains. One is the S type and second is the R type. S type, as the name indicates, S stand for the smooth. So the outline, or you can say the texture of such type of bacteria, it is rather smooth. So the name is given smooth. It is virulent and pathogenic. That means it spreads the certain type of the disease. Here we are related with the pneumonia. And this type of bacteria, it is encapsulated. That means the capsule is coating or coating is there. We know that the tablets and capsules, same meaning is here. Capsule that is having the outer coating, which is a smooth one. So it is encapsulated. So this is the first strain, that is the S strain, which is caused the disease pneumonia, and it is a pathogen. Second type of strain is called as R type. R, again, the name indicates R stands for the rough. So it is not a smooth, but the coating is rough and it is non-capsulated. Capsule is absent. As well as it is a non-virulent and non-pathogenic. That means it is not cause for the pneumonia. So it is we can say this type is non-pathogenic, harmless in short. So these are the two strains he studied. And as I told before, he chose some group of the mice and he injected these strains one by one. So one after one, he injected such a type of the different strains and he observed certain results which are indicates or which gives idea of which is the factor that is responsible for the pneumonia. So these two types, R type, let us start with the R type. So the first, he injected this R strain to the group of this mice, group of mouse, that is a mice, and he injected to that group, and after some days, there were observation. He observed that they survived. That means without any type of the disease or the pneumonia, they survive. They remain healthy in short. So this is the result of R strain injected into the mice. While in the second experiment, in the second group, he injected S strain, which is a smooth, pathogenic, encapsulated, and virulent. So this S strain, when he injected to the mice, he observed that after some days they suffered with the serious disease of the pneumonia, and the mice died. So the death, it is because of that pneumonia, because of S strain, in short, which are virulent or pathogenic. So this is about the two sets. Now he performed this experiment for the third type of the set of the mice, and what he did, see here that the same type of the strain he utilized, but it is treated. That means he hit and killed this S strain, which is responsible for the pathogen or the pneumonia disease. And he found that this third group of the mice survived. That means without any type of the pneumonia, no suffering. And these mice were he found healthy. So these three experiments, they denote us R is not responsible, of course, but S is responsible for certain type of the disease, that is the pneumonia. But when he treated that S strain, that is a heat-killed S strain, and he found that whatever the contents which were responsible for the pneumonia, they become inactive or it killed him, in short. And he found that the mice, all the mice were survived without any infection. So these are the three experiments. And the last experiment he performed that he mixed these two, that is the heat killed, that is the third experiment, as well as the second. Third, that is heat killed S strain mixed with the R strain. And he found that the mice again were suffered with the pneumonia and died. So this is very miraculous or the explainable thing that S, which is pathogenic, must consist something. But when it is treated, heat killed, then it becomes inactive. But when it is mixed with the R strain, it again become active. That means something present in the S that goes to the R strain, and R strain now become to the S strain. So let us see with the help of some figures or diagrams that is more convenient for you to understand. So living S strain, it is virulent, as we have studied here, that S strain which is responsible for the pathogen, that type of the disease. And when he injected here, you can see here syringe and this is the mouse. When he injected, he found the result, that is the mouse died. In the second experiment, R strain, and having the same type of the treatment, that means R strain which is a non-virulent, when injected to this mouse, and he found that the mouse lives, that is without suffering of this pneumonia. Okay, so these are the two strains. Now the third strain, where heat killed S strain, which is responsible for the pneumonia and cause of the death, he observed that when it is heated, heat killed type of the S strain, the result observed, it is similar to that of the R strain. That means what is present inside the S strain now killed and does not affect. It is not cause of suffering of any type of the disease like the pneumonia and that group of the mouse, that is a mice, that lies. So this is the interesting thing, that the S strain, when it is heat killed, then no effect on the mouse, but that lives. While in the fourth experiment, now there is a mixing of these two, that means R mixed with the S. So these two, they are mixed, and when they are injected to the mice, he found the same result, that is of which was in the S strain. So here the things are very interesting, that something was there which remained alive even though heat killed, but it is transferred to the R strain, and the result is that the same one, it is with certain type of the conclusion that the mice were it effect, and this R strain that accept from the S strain which is already hit and killed. So R is converted into S, so that something that accepted, that principle accepted. So the Griffith experiment proved that something is there in the S strain, even after the heat killed treatment, it survives, but does not affect to the mouse. But when it is with the R strain, it is goes to R, and the R now, which is harmless, it is converted into S strain, and this effect to the pneumonia disease in the mouse. So in this way, we can say that the conclusion of the Griffith experiment is that the R strain must have taken up, that is picked up, that particular factor, that is the transforming principle, and which allowed R strain become virulent. So R is converted into S, and it shows the effect which was shown by the S type. So in this way, Griffith experiment that conclude where the same type of the result which observed when the simple S strain injected to the mice. So in this way, this is the Griffith experiment. So many times it is asked in the exam. So you must know about these things. You must remember about the name of the bacterium, Streptococcus pneumoniae, then S type, what is S, what is R type, and then the experiment of the Griffith, how he injected, what are the results. In this way, this Griffith experiment may be asked in the exam. It may be for the 3 marks, and you must know about the all things which we have studied. It is very simple, but just you have to remember these 3 things. That is the name of the bacterium, S type, R type, and what are the steps he followed. You must remember. So in this way, this is all about the Griffith experiment. Now, the next experiment that is based on the same, that is the Griffith experiment. What are the results are there? The group of 3 scientists, that is the Ivory, Mccarty, and Mcleod. So it is known as the Ivory, Mccarty, and Mcleod experiment, where he isolated different contents from the blood. In the first test tube, you can see here DNA, deoxyribonucleic acid, then RNA, ribonucleic acid in the second test tube. In the third one, that is the proteins. So these are the 3 contents he observed, and he isolated these 3, and they are mixed with the heat kill S strain plus R strain. So this is the combination he added in all these things to find out which factor is responsible for the carrying the information or the genetic principle. So in the first test tube, where the only DNA, he observed that this pneumonia, that is the Streptococcus pneumoniae, this type of the bacterium, they are converted and they become smooth walled. The strain is smooth. So R is converted into S. That means R accepts something from the S. So R is converted into S. That is the transformation. While this DNA, he found that they are accepted and transferred. There is a transformation takes place. But in the next 2, he found that RNA, same, that is the no change in the RNA, it remains rough type. You can see here, this is the R type, that is a rough type. And the proteins are also isolated, where remain at the same one, which the hit kill S strain and the R strain. So both the things, that is both the test tube, RNA as well as the proteins, there were no transformation. And only DNA, that is the test tube, get transformed from R to S. Means DNA is responsible for such a type of the transformation or the genetic principle. For to conclude this and for to collect the more evidence, what he did, this DNA when it is added with the certain type of the enzymes known as the DNAs. So DNAs are the proteins or the enzymes which digest this DNA so that it become inactive. That is DNA become inactive, digested. So DNA is enzyme when they are added in the same type of the test tube and same result, that means just like the RNA and protein. So this cannot be converted and remain the transformation without transformation. So no transformation in all these three. So this is the uh, certain type of the conclusion that DNA is responsible for the transformation. But when DNA is digested with the help of some enzymes, DNAs, which break, you can say you can break the DNA by adding the DNA's enzyme. That means it become inactive, and when they are inactive, there is no transformation. So in this way, this Ivory, Mccarty and Mcleod experiment prove that certain type of the DNA, that means only DNA and DNA is responsible for the conversion, that is called as the genetic information material. And hence, they concluded that every Mccarty and Mcleod experiment that prove that the transforming principle is nothing, that is not RNA, not proteins, but the DNA. So this DNA, especially in the eukaryotes, we are talking about, this act as the transforming principle. And hence, the conclusion of these three scientists, which was based on the Griffith experiment, they concluded that the transforming principle is not other than the DNA. And hence, we can conclude that DNA is the genetic material and not proteins or RNA in eukaryotes by using the bacteriophages. So in this way, this experiment gives us the clear-cut idea and the conclusion that DNA only the factor responsible for the transforming principle or the genetic informative material which is responsible for the transforming from one generation to the next. And hence, such a type of the experiments that gives us the idea and clear-cut idea about the DNA which act as a genetic informative material. Now, the further experiment are there which also having solid evidence related with the DNA as a factor transforming principle, that is the Hershey Chase experiment, and they utilize the radioactive substance, that is using the phosphorus as well as the sulfur. But we will deal that to the next lecture. But for the timing, you must remember that these are the two experiments and may be asked in the exam, and they have certain type of the result, that is the DNA, which gives us the idea, clear-cut idea. But the next experiment, that is the Hershey and Chase experiment, gives us a solid evidence, and hence we will see that to the next lecture. For the timing, we will stop here, and thank you.